Right. Our next speaker is Dr. Stephanie Brown with a talk, Ultra-High Resolution Imaging of Amygdala Subnuclei, Structural Connectivity in Major Depressive Disorder.
Okay. Hi, everyone. It's great to be here today, and I'm going to talk a little bit about some of our 7 Tesla work, and that is ultra-high field resolution imaging of the amygdala subnuclei structural connectivity in major depression disorder. So a bit of background about what makes amygdala connectivity relevant to studying major depression. We know that the amygdala is really important in modulating emotional context and processing, and we also know that it's been implicated in lots of different mood disorders such as PTSD, bipolar, anxiety, OCD, and lots more. The current status of the literature is that seed-based resting state fMRI usually pretty robustly shows widespread decreases in whole amygdala connectivity, but there's also certain regions where you see increases as well. And many of these measurements are associated with severity and anxiety, severity of symptoms and anxiety. But to date, there's been much less investigation into the structural connectivity of the amygdala in MDD, and the individual amygdala subnuclei, which are structurally and functionally distinct, they haven't been examined to date. So classically, the 3 main white matter efferents from the amygdala are the stria terminalis, which is thought to originate in the central nucleus, the anterior commissure, and the amygdala fugal pathway, which runs through the basal, lateral, and central nuclei. The anterior commissure couples the amygdala hemispherically, and it's really involved in emotion, olfaction, instinctual behavior, and memory consolidation. The amygdala fugal tract is thought to exert downstream effects on the hypothalamus and the septal nuclei, and therefore it's really important for influencing threat reactivity. And the stria is really more involved in social conduct and motivational conduct. The other important white matter connections for the amygdala are the uncinate, which is the main connection between the limbic lobe and the frontal cortex. This is a late developing tract, and it's pretty susceptible to psychiatric illness, probably for that reason. And it's more involved in self-awareness and declarative memory. It can be thought of as a more higher processing white matter tracts. And then there's also reciprocal visual connections with the amygdala in visual cortices 2 and 4 and the superior colliculus. And this is thought to be linked to a coarse threat response, which facilitates unconscious selective attention switching. So using our increased resolution at 7 Tesla and a recently developed algorithm by FreeSurfer, what we can do is segment the amygdala into its constituent subnuclei. How we did this is we utilized the T1 weighted MP2 range and the T2 weighted TSC image to put the images into a multispectral segmentation, which leverages the increased resolution of the T2, even with its anisotropy, and we get a pretty reliable segmentation. In the middle row, you can see quite clearly the larger nuclei, so that is the basal, lateral, and accessory basal. And then in the bottom right hand image, you can see that those are the smaller nuclei, so that's the central, medial, and cortical nuclei. So to look at the connectivity, we used a diffusion-weighted sequence. We used MR-Trix for this, which uses a constrained spherical deconvolution to estimate fiber orientation distribution, which is also known as the FOD image. The tractography was performed probabilistically. It was seed generated, and an FOD amplitude threshold of 0.15 was used. So that's quite stringent. So we don't expect to see many spurious streamlines, and then also a threshold angle per voxel of 60 degrees. The amygdala nuclei ROIs were randomly seeded within the mask without terminal or inclusion ROIs, so basically to whole brain. And we had an enhanced resolution of 1.05 millimeters isotropic for this data, so that's better than the average. What we saw is that in each participant, the 5 tracts that we expected to see were pretty reliably tracked. So we see the stria, the uncinate, the subcortical visual pathway, which appears to pass along the inferior fronto-occipital fasciculus, the amygdala-fugal tract, and the anterior commissure. So we can visualize this tractography as both coming from the whole amygdala and also coming from the individual amygdala nuclei. And I've circled, because it's not quite clear, where the amygdala regions of interest actually are in this diagram, and this is in an axial view. So qualitatively, virtual rendering of the amygdala subnuclei shows 2 important points, which is the nuclei exhibit differing profiles of streamlines, and the basal and lateral nuclei appear to exhibit shorter range connections than the medial, central, and cortical. It's really important to bear in mind, however, that the methodology can't actually accurately differentiate between emanating fibers and fibers passing through the regions of interest. And this is just important to bear in mind because the nuclei are highly interconnected. So once we have produced the tractograms, we can then use the microstructure scans to do a quantitative assessment of the tissue. And this is a fractional anisotropy map. And one of the things we can do with this is we can also get the mean diffusivity, the axial diffusivity, and the radial diffusivity. And these are all slightly different measurements which can tell us things about the integrity of the tissue. It's really important to also caveat this, however, that interpretation is limited. And with tractography, things like crossing fibers, etc., can cause changes in the microstructural measurements that do not necessarily equal things like axonal degeneration. So for each streamline, in every voxel and every streamline, microstructure measurements were taken, and then a mean per streamline was created, and then a mean per bundle. And then we took these measurements forward for statistical testing. And what we saw was that there was, in many nuclei, results that suggested an increase in microstructural order, which can loosely be interpreted as increased connection strength. All of these except one were in the right hemispheric nuclei, especially the basal and lateral nuclei. The accessory basal was pretty much unchanged. There were changes in the centrocortical complex, and these were driven by the central nucleus, and all of these survived false discovery rate correction. So in the left hemisphere, we also saw that there was evidence for decreases in microstructural order, particularly at the basolateral complex, apparently driven by the lateral nucleus. One of the interesting things that we can do with this tractography method is we can apply a technique called spherical deconvolution-informed filtering of the tractograms, which removes streamlines that are very unlikely to be biologically relevant. So when we do that, we then are left with a streamline count that we can interpret as connection density. And what we saw in the left medial nucleus was that there was a decreased connection density in major depression. We also saw that there was an increased connection density in the left hemisphere at the lateral, basal, and central nuclei, and also the centrocortical complex. So one of the things that we can follow on from this analysis is to manually isolate each of these 5 tracts, the stria, anterior commissure, amygdala-fugal tract, the visual connection, and the uncinate, to try and tease out which one of these particular white matter tracts are contributing to these significant changes in MDD. So to kind of summarize, our findings are consistent with functional and diffusion findings that have been reported previously. And what we see is that the amygdala is structurally hyperconnected in major depression in the right hemisphere. And this might be predicted given the amygdala's really prominent role in processing of negative emotion and regulation of the HPA axis. We also see pretty profoundly that the right and left hemispheres exhibit different changes in connectivity in MDD. when we look at the amygdala nuclei as separate entities, we show for the first time that the nuclei actually appear to be differentially affected in terms of their connectivity, which might be really useful in the future for informing us about their relative roles in mood disorder and emotion. Thank you.